Jason Climer will be speaking to us today, and I have a bio for Jason. He received his bachelor's degree in biology and biotechnology with a concentration in cellular and molecular biology and genetics with a minor in computer science from Worcester Polytechnic Institute in 2010, where his work on neurotransmitter receptors in the worm C. elegans received the Provost Award for Most Outstanding MQP senior thesis. He did his doctoral work in the lab of Professor Michael Hasselmo at Boston University, where he published several papers on his studies into the role of theta oscillations in memory and navigation, and he is now a postdoctoral fellow in the Dombeck Lab at Northwestern University. He is a hobby artist and science outreach enthusiast and has created the YouTube channel Garden Variety Science to promote science. Jason is also a gardener, biker, and the father of those adorable twins. You just heard them up there in the balcony. So, Jason, welcome.
Thank you all so much for inviting me to talk here today. This has been such a welcoming community to come to since we moved to the Chicago area. And I was really excited to talk to you guys, and but I needed to think about what I was going to talk about. There was a lot that I could cram into this talk. I want to talk about my own work, but before I get there, I want to talk about science in general. So I sort of started my talk by thinking about what is science. I think everyone in this room probably has a really good idea of what science is. And so I'm not really going to go through the nuts and bolts of the scientific method, but I am going to talk about what science is to me, what I think it is to society, and how I think the way we talk about science isn't particularly productive. So who here has seen a diagram of the scientific method that looks something like this? Anyone? Anyone seen something like that? Here's another one. This came from the same Google search for scientific method. This one's in Russian. We've got a nice little circle here. We've got, this one's a branching flow chart. We've got branching circles and all this. I actually kind of like this last one, but the point of all this is that we don't teach what the scientific method is in a consistent way. And the reason is that science is a little bit more complicated than a simple flow chart. And I think the best place to start, if we're really going to talk about what science is to talk about science as a philosophy. I have a PhD, I have a doctorate in philosophy, and the reason for that is that science is the natural philosophy. It is a way of thinking and about the logic of the world that we can use in a really practical way. So science really got its roots in the ancient world. So it has roots in Eastern thought, in Middle Eastern thought, and in what many people know, the Greek origins with Aristotle and all of that. And really, after the Renaissance and the end of the Renaissance was characterized by what is called the scientific revolution. And this is where sort of the modern way we think about science really as a global community got its start. Science is a subset of epistemology, which is the focus of the Sunday school this year, and so I wanted to give a little bit of a touch on this. It's the philosophy of knowledge. How do we know what is true or what is not true? As a really simple example of this, I'm going to start with René Descartes. René Descartes was a mathematician and a philosopher, and he's most famous for these words that he wrote in a treatise on epistemology. I'm going to have terrible French, so I'm actually just going to not even try this time. But translated, it is, I think, therefore I am, which almost everybody has heard. The idea behind these lines is that he said, all right, I'm going to take everything I know, everything I think I know, and I'm going to doubt it. And I'm going to put it, furthermore, I'm going to posit an evil wizard. And this evil wizard is trying to fool me everywhere I look. If I look out the window and I see a sunny day, it's actually not a sunny day. He's made it look like a sunny day. So how can I know what's real and not real? His argument from there is really unsatisfying to a humanist. From there, he starts to argue that there is an almighty power that is all good and therefore wouldn't allow such a wizard to fool him. And so this is not the approach that science takes. Science still does doubt everything, but we're going to do a little bit of a finer grain division of it. So in addition to being a scientist, I am a statistician. I've done a lot of work with statistics. It's my favorite field of math. The reason why I like it is that it's the math that revolves around uncertainty. So if we took every idea that could be true or not true, and we put it in one big bag, a statistician might draw something like this, a big circle with the letter omega in it. And so how am I going to tell what things in this circle are true or not true? Well, we're going to start by applying a logical device called Newton's flaming laser sword. This is a logical device like Occam's razor, where we can take ideas and we can separate the ones that are likely true or likely to not be true. In this case, we're not doing true or likely not true. Michael Adler, who invented Newton's flaming laser sword and gave it its really catchy name, said, what cannot be settled by experiment is not worth debating. Now, I think this is sort of a harsh way of putting what the philosophy of science does to ideas. But I think a better way is that if it can't be settled by experiment, then we can't tell if it's going to be true or not true about the physical world. Because if we're doing science, what we really want to do is find out anything that is true about the world that we can settle by experiment. So we'll take all of our ideas, we'll apply Newton's flaming laser sword, and we'll get rid of a lot of them. We're just going to ignore most of these ideas for now. It may become true in the future that technology or something will allow us to test these ideas, but we can only work with things that we can test experimentally. Now, what does it mean to test something experimentally? It means I have an idea that predicts something when I do something. If I'm going to look somewhere, I'm going to expect to see a certain thing. Or if I'm going to change something in a system, I'm going to expect a particular outcome. Now, if that outcome doesn't happen, then I know my idea is false. Now, we often think about science as being something that is true. And science is typically not done in a fashion where we're affirming the truth of things, because it's really hard to do that. The classic example of this is a black swan. If we use the statement, all swans are white, this is a very difficult thing to assert if you're going to say it's absolutely true. It's easy to prove it wrong. All we need to do is find one black swan. But if we want to prove the statement every swan is white, we have to look at every single swan. So we can quantify our uncertainty about things using statistics now. But this is still the way we do science, is that we take an idea. If you love an idea, the best thing you can do in science is try to prove it wrong as hard as you can. And so what we're going to do is we're going to prove ideas wrong that can be tested by experiment. We're going to prove more and more ideas wrong until we have a very narrow explanation of the world. And this is how science works. Now, this takes a lot of work and a lot of time. There have been hundreds of years and hundreds of thousands of people working on these problems. And this is going to go across lifetimes. So how do we know that we can trust this if we're not seeing things with our own eye? This is a thing that often comes up when people are arguing against evolution and other big ideas in science. They say that you weren't there, you haven't tested this thing directly, how do you know? The answer to that is, the answer that we've come up with as a civilization is peer review. So what we have done for science is when someone comes up with an idea and tests it with an experiment, before it gets disseminated to most scientists, it goes to a narrow set of experts in the field. And they either can decide if the experiments actually show what the authors claim they're showing or not. And this precludes being published. And so this is really why we trust scientists. This is why, because the scientists don't trust themselves. They are trusting each other and the community. Now, the peer review system is not perfect, and it currently is under a lot of stress by the modern state of science. But I'm going to talk about all that later. But for now, I really want to come back to a couple pithy quotes about science that I think kind of misrepresent science in a little bit. This is a really famous quote about science from Neil deGrasse Tyson. The great thing about science is that it's true whether or not you believe it. Now, I have a ton of respect for this guy. I think he's done really great things for science advocacy. But I slightly disagree with the word true here, because we're not proving anything is true. We're really just taking falsehoods and uncovering them. And we do that enough we can find out what is really true about the world. Perhaps a better phrase, and I'm going to use the word models a lot in my talk. I'm going to talk about models as what we often talk about as theories or hypotheses. Because what we do when we're trying to explain the world is we make a simplified rule that we think explains the world. Now, that rule is probably too simple. Humans are dumb monkeys compared to the complexity of the universe. So George Box, he was one of the pioneers in modeling or taking computers and doing simulations of things that are parts of the brain. And he said that all models are wrong, but some are useful, because he knew that none of his models really well explained how the brain works, but they could do things that are interesting. So how useful is science? What has it done for civilization? And this is a thing that I have just an incredible amount of awe for, because just by the numbers, science has just been one of the most impactful things, perhaps the most impactful thing that has ever happened to our species. So if you look in ancient Greece, and if you look at the state of medicine, this was the height of medicine in the Western Asian world, there was still, most of the medicine was based on trial and error and based on old wives' tales. With science, we soon found out that we're covered with microscopic things that cannot be seen with the naked eye, that can cause disease, but also can help us. And this is something that for the vast majority of human history, we had no idea. Diseases are not caused by evil spirits or by a capricious god, but by an organism. And from that, we've been able to improve our food, the way we make our foods, and the way that we do medicine. And that has led to a huge population explosion in humanity. In fact, the scale of the population explosion that's been going on is unlike anything that we've ever seen. Until 1,000 years ago, there were less than 1 million of us, and now there are almost 8 billion of us. And this is just a huge, huge difference. And we owe science to this for a lot of this survival. Another great thing about, another thing that has happened because of science is we've gotten computing power. This has really made it so that the science that I do is a lot easier to do. And it's also been necessary for a lot of our modern economy and for dealing with the large population that we do have. Early computers were no smarter than a calculator, and they were huge, very expensive, but because of things like the space race, because of things like the Cold War, these things caused the government to invest a lot in physics research and computer science research, and we developed technologies at an extraordinary rate. So we've gone from a large machine that can barely do basic math operations to your phone, which is more powerful than any of the early supercomputers could even dream of. Our supercomputers now are truly something to behold. They can do millions of operations. They can store lots and lots of data, and people are now able to purchase these portions of these computers, time on these computers, from big companies like Google and Amazon to do science with. In Europe, their version of the BRAIN initiative is the BRAIN project, and they're trying to literally make a computer simulation of a human brain. We're just getting to the part where the numbers are starting to match in terms of having the computational power to do that. If we look on the practical side of this, with most computers, though, are much, much smaller than that. We can't get anywhere close to what the human brain is. But even with a smaller neural simulation, we can do some pretty interesting things. So Google has been trying to use neural networks to recognize objects. And this rather interesting picture here is it looks kind of artistic. What it is an iteratively changed picture. It's been called Google Deep Dream, where it tries to recognize things in the picture. It takes what it recognizes and just loops it over and over again within the algorithm. Perhaps in a more practical sense, these networks have been used a lot for recognizing objects. And you can actually, a recent paper showed that if you take a neural network, you can train it to take real photos and paint them like Van Gogh painted them. This network can actually also take paintings that were done by Van Gogh and turn them into photorealistic images, which I find absolutely incredible. So we're starting to do some really wild things with this. And one of the really strange things about it is we have no idea how these work. The best mathematicians in the country are now working on these problems, trying to look at these computer programs that do work, but we just don't know how they work. And so this gets into the next part of my talk. What is neuroscience? So this is the field of science that I'm in. I'm really interested in how the brain works. So we can kind of append that question by Descartes. I think, therefore I am. What am I? I am mostly a thing that lives in my skull that's right between my ears. That's about the size of a loaf of bread. Compared to other fields, neuroscience is a relatively young science. This is Ramon y Cajal. Ramon y Cajal is thought to be the father of modern neuroscience, and he's a lot of actually really interesting writings on being a scientist and the science's role in society, but he was also an incredible artist. In fact, this is probably where his talent really lied. So at the time that he was doing research, the going thought was that the brain was one giant syncytium of fluid. There was a lot of little tubes in it, but the fluid would move around in your brain, and this is what let you think. Ramon y Cajal, for the first time, was able to stain individual neurons in the brain. And when he was able to do that and draw them and reproduce them over and over again, he came up with what is known as the neuron doctrine, that the brain is made up of distinct single-cell units called neurons, and that these neurons send long projections, these long projections, these long bodies that go out of the cells, and these are called dendrites, and they match up with axons, and then communication can happen between these tiny units. Now, this idea was laughed away when he first proposed it, and one of the reasons is that the corollary of this is that in order to understand the brain, we have to understand what 100 billion things are doing. There are 100 billion neurons in the brain. This is an astronomical number, like literally an astronomical number. If we want to look at how many 100 billion are, let's look at it if we had 100 billion $100 bills. So now this is going a little fast. So we've got 1 and 100 here. This is what 10,000 $100 bills would look like. I'm just going to say dollar bills instead of $100 bills because they're the same size. This is 10 million, so now you can start to build furniture and sit on it. But we're nowhere close to where we're going to be. This is 3 billion. So you'd have to move it around in many trucks. And this is 100 billion. It's basically a city of dollar bills. And dollar bills aren't that big. And this is only part of the problem, because that's how many cells there are in the brain. These brain cells each have about 1,000 connections with each other. And this is what you really have to understand in order to understand the computation that's going on, is what are all the inputs onto the cells. That means there's 100 trillion things you have to look at the brain in order to understand what it's doing. This is an enormous, monumental task to go at. But it is something we are trying to do. So when I was doing my PhD work, which is mostly what I'm going to talk about when I talk about my own science, I was doing extracellular recordings. This is one of the most prominent techniques over the last 100 years for learning how the brain works. So what we have here is we have an electron micrograph of the kind of electrodes I used mostly. These electrodes are 4 wires that all together, when they're bundled together, are only about 50 microns wide, so which is much thinner than a human hair. And we can lower this into living brain tissue and get electrical signals from the end of it. And so this is a cartoon of what that looks like. So at the bottom, we have a bunch of neurons in a layer right here. And we've got our 4 electrodes coming down. And because these electrodes, even though they're so small for us compared to neurons, they're relatively large. They're different distances from each neuron. And so when a neuron fires, you can think of it as surround sound mics. We can listen for where the different neurons are, and we can tell them apart. Now, this can only get you at most about 100 neurons at a time, though you can get a different signal, which is a brainwave signal, from about an area that has about 1,000 neurons in it. And that's something I might call LFP or theta interchangeably in this talk. But I'll explain that again when we hit it. But this is, so now we're going to start talking about my science, like really the science that I did to get my PhD, which is kind of exciting. I worked in the Hasimo lab at Boston University. Michael Hasimo was my PI. This is a term that you might hear if you start to research how science is funded. This is the principal investigator. This is the person who runs the lab. It's usually a professor. This is sort of the goal for my career, would be to be a PI. I recorded individual neurons and brain waves, or theta, from mice and rats, and I did experiments and analysis using this data. So I measured a thing called 2D phase precession, which I'll talk about in a little bit, in cells that are cells called grid cells, which are one of the most remarkable discoveries in this area of the brain, or any area of the brain, in the last 50 years. And I also developed new ways for measuring the effect of brain waves on single neurons. Now I'm working at Northwestern University using calcium imaging, which I think is the technology of the future for really trying to get at these larger scale things that are going on in the brain. So in the title of my talk that went out, I said that I was studying memory. Very little of my talk is actually going to be about memory directly, but I want to talk about why what I'm researching and memory are linked. And the discoveries that have led to this have really changed the way I think about problems and complexity in general, and also really changed the way we think about memory, how memory works. So this is Henry Molaison. He has probably made more contribution through his work to neuroscience than any other human being. He died in 2008. He was not a scientist. He had a bicycle accident when he was a boy, and following the bicycle accident, he had intractable epilepsy that couldn't be treated with any drugs. His epilepsy was getting worse and worse, and it was going to kill him. Dr. Scoville, his surgeon, his neurosurgeon, suggested doing a radical surgery. They were going to remove both of his medial temporal lobes. Now, the medial temporal lobe is basically straight behind your eyes in the center of your head. It's really hard to get at. It's one of the most protected areas of your brain. It very rarely is damaged from injury. It is also one of the areas where seizures do come from more than any other part of the brain. But we've become a lot more selective in how we try to treat epilepsy in these regions since patient HM. Because once we removed both of his medial temporal lobes, the hippocampus, which is a structure we now know is incredibly important for memory, he was never able to form a new memory. There's some really incredible NPR footage out there where they did an interview with him talking about how he can remember things. He knows he can't remember things, and he remembers things from before the surgery, but he never formed a new memory. It's a very selective deficit. So this is great. We know what part of the brain is important for memory. So let's take an animal model and let's record from it. Let's see what these cells are doing. So if you put an electrode in the hippocampus of a rat, the first thing you see is a brain wave. It's a big brain wave. It's one of the most consistent brain waves in the rodent brain, and it's called theta. It's about 8 hertz. This is what it looks like. And this phenomena is a little bit controversial about what its role in memory is, but it definitely exists in this area. If we start to look at the single cells, we can see something that just blew my mind when I found out about it. And it's been so pivotal to our understanding of the brain that it was awarded the Nobel Prize in 2014. John O'Keefe, Mabry Moser, and Edward Moser shared the prize. So here is a track from a rat. The rat is running around a box looking for bits of Fruit Loops. They love Fruit Loops. And as he runs around, you're going to see little red dots appear on the screen. These are the action potentials, the communication signals sent out by a single neuron. I sped this up so that it'll cover the entire box, and you'll notice that the cell fired only when the animal was in this portion of this corner of the box. The hippocampus is full of these cells. You have cells in your brain right now that are firing just because you're here at the Ethical Humanist Society, or even on the couch back there. And this is the area that we use memory. So what is a place signal doing here? Since then, we've now realized that our memories are not an ephemeral experience. They're actually a map. This is something, the most prominent idea of how memory works these days is that you use the place where you are, the different spaces that you're thinking about, like time we would consider a space. These things are really mapped onto these cells, and that's how we structure memories. Now, I think these cells are fascinating, and they're now the kind of cell that I work with the most. But when I was in Dr. Hasselmo's lab, I was working mostly with another kind of cell, which is even more bizarre. So these are one step upstream of place cells. These are grid cells. And so this is, again, a similar video, except this is, you can see the lights on the apparatus that we use to record the neurons on the animal's head. As he runs around, you can see those lights, and we're just, again, leaving little red dots where this neuron fires. This one maybe have been looking for Cocoa Krispies. So he zooms really fast, and you can see that this cell is firing in multiple places in the environment. But what's really interesting is that these locations that the cell fires actually fall on the corners of tightly packed equilateral triangles. In your brain, there are cells that will make a hexagonal grid across the entire world. This is what made me jump into this field head first. I really wanted to figure out how these cells work and why we would ever do that. And we still really don't know. It's very controversial. This is bleeding edge research. But one of the really interesting things about this brain rhythm and the firing of these cells is that they seem to be related. So this brain wave, theta, acts as a clock for the entire network. So if we have an animal who's, I'm going to move over here a little bit. If we have an animal who's running along and running through two place fields, so these are areas where a neuron in his hippocampus will fire. If we look at that neuron, it'll start firing relatively late. Let's say it starts firing at around 11 o'clock. We'll call this peak 11 o'clock. And as he goes further and further through the field, that firing will come out at earlier and earlier times. So we can actually just read out from a single place cell how far the animal has gone through that place cell's field. And what happens as a result of this is if these fields overlap, on every cycle of theta, you get a sequence where it plays out cells from behind that represent places behind the animal to places in front of the animal. And this is happening 8 times a second in the brain of a rat as he runs around in an environment. Now, I think this is really cool. I think this is a really interesting signal, and I have no idea how the brain does it. We know that this happens in grid cells as well. So this is a grid cell that actually I recorded. But this is a rat running down a linear track here. And as the animal goes down the linear track, I'm going to show some plots that look like this. It's okay if I'm not going to go into too much detail about this plot, but on the x-axis we have the position, and on the y-axis we have the theta phase. And so again, this is going up and down with theta, and so that you can think of this as the time on the clock. And as the animal goes through the field, you can see this structure, this sloping structure here, and that's what we see phase precession as in grid cells. Now, it's really hard to examine these cells as the animal's running around in the open environment because the animal does a lot of random stuff. He's looking for cereal all over the place. He swings his head back and forth constantly. And then rather than being able to analyze like on the linear track where the animal is restricted to going in just a narrow area, he can go anywhere he wants. And this will become important in a second. So there were some people that came up with ideas of how precession might be generated. And so here's a little toy model I built that explains this. So this is what we call a temporal interference model. So what we're going to do is we're going to start with 2 oscillations that are completely out of phase. So these are 2 signals that are just, they make a wave. And when they're out of phase with each other, they'll destructively interfere. They'll cancel each other out. Now, we're going to start to move this animal towards the center. And as we do, we're going to shift the frequency of one of these oscillations so that they start to come into phase with each other. As they come into phase with each other, they'll add constructively instead of destructively. And so you get an increase in the amplitude of the output. Now, one of the interesting things about this is that just by using these two signals, we can generate phase precession. If you look at the peaks of this versus the peaks of this purple line versus the peaks of this gray line, the peaks come earlier and earlier as the animal moves further through the firing field. Now, this does a great job of explaining that stuff on the linear track, but you have to do some weird things in order to get these models to work for grid cells in an open environment. And so when I first joined the lab, I messed around with these models, and I tried to get some predictions as to what these models might think is going on with precession in the open field. Again, I like this model. I think it's elegant, but I want to prove it wrong. If I can try to prove it wrong, that's just evidence for it. So here's the output of my model. So I've got a virtual rat running around a box, and he's going in and out of these firing fields. So we have a cell that's firing some when he goes into the field and stops firing when the animal goes out. This blue line goes up and down as the animal goes in and out. The higher this blue line goes, the closer the animals come to the center of the field on that particular pass through a field. But if it goes a little bit shallower like here, the animal's just gone around the edge of it. And the model predicted something pretty striking to me. So this is the phase that the model predicts. And if we look at this shallow pass versus this steep pass, so this pass that goes to the center versus this pass that goes around the outside, we can see we get full phase precession in both cases in the model. This seemed like something I could look for in real data, but it took me a while to figure out how, so I actually developed a whole new technique for looking at phase precession. So this is, which I won't go into all the details of now, but this is that nice structure that we saw looking at these open field environments. And I could ask that question. I could now take just the passes that either clip the outside of the field or go through the center of the firing field, and I could see whether they have different structure. And they don't. We couldn't find any a difference in the structure. And this has been corroborated by a couple other groups now. So I feel fairly confident that at least this behavior of the grid cells matches that model. Now, there's been a lot of other evidence that has brought this model into question. I don't want to say that it is absolutely what's going on. This is definitely a point of contentious debate right now in my field. And so for the next part of my talk, I'm going to talk about the second project I really did that was looking at some of this evidence that suggests that maybe these oscillations aren't important for making grid cells. And for that, I'm going to first show this fun little video of a bat stuffing his face with a banana. Because we just took a real deep dive into neuroscience, and it might be nice to look at a bat. So, bats are really interesting when we're starting to look at this, because one of the things we're now thinking is really important to memory is that an animal is navigating. So a fruit bat has a remarkable ability to navigate. They actually learn where different trees are that are good sources of food, and they can do something that rats can't. They can fly. So we can start to look at how animals can navigate in 3D environments. You can train a bat to crawl around in a box, and a group did, and they recorded grid cells in these animals. And so this is what their grid cells look like down here. One of the striking things about these recordings, though, is that they weren't able to detect theta oscillations in the firing of these neurons. Now, if theta oscillations are important for making that firing pattern, and there are grid cells but no theta in bats, that is a big strike against that kind of model that I was talking about earlier. In their paper, they have this quote. This is actually the last line of their paper. More generally, we provide here a rare example of causally disproving a major class of computational models of a higher brain area. Now, this was a fairly controversial statement to make, especially from people that think that oscillations may play a role. And the biggest criticism against this was that the theta rhythmicity was not the only difference between bats and rats. So this is a rat grid cell, and this is how we usually measure the rhythmicity of an individual neuron. Briefly, what this is it's the probability that you have a spike, and a spike again that many milliseconds later. If a cell is rhythmic, Though what you should see is that this looks like a nice wave. If a cell is not rhythmic, this is flat or just kind of a squiggle that doesn't make any sense. This is the same cell, but and so it should be just as rhythmic. But what we've done is we've corrupted it. We've just pulled out random events from for random spikes from our data set. And the result is a cell that still looks kind of like a grid cell, but we don't see any of this nice wave in the autocorrelation. So this is a rebuttal to that original paper, and with good reason, the original authors of the paper said, well, this is not nearly thorough enough to convince us that this is a problem. Now, so what I wanted to do was to be that thorough. So that was the next project that I worked on. So I did a lot of work on this project, and I'm not going to be able to go through it all in great detail, but what I'm going to quickly show here is a similar analysis to what we saw with that one rat grid cell. This is simulated neurons, though, so now I know for sure that every one of these neurons is exactly as rhythmic as the other ones. And the only thing I've changed is how much that cell fires, how much data we get out of that single cell. And you can see that just by reducing that firing rate, I can really reduce the score that we get for how we measure theta rhythmicity. And this is a big problem. What we really like is if we have less data to not have a bias in this measurement. We'd rather have it just be not as reliable in terms of consistency. I developed a new technique for looking at rhythmicity that is much less hindered by this problem, but it should be noted that no matter what, if you don't have enough data, you cannot say whether or not a thing is true. Absence of evidence is not evidence of absence. And so here are autocorrelograms from those bat grid cells. I've got four here, and this is the best fit that I could do of this autocorrelogram. And none of these look particularly wavy, and the statistics work out that none of these are significant. But I can also do the statistics and say that there's just not enough data here to really say one way or the other. So I think that that particular data set is just not very good for looking at this. So in summary, we had the big question of whether or not there was theta rhythmicity in bats. And my answer is, I don't know. Specifically, I think that the data sets that we've been looking at to try to answer this question just aren't sufficient for doing that. And there are other data sets out there that might be better, but there's a lot of caveats when you're doing these kinds of analysis. Now, a bat does not move like a rat does. When a bat is crawling, which is not a natural behavior for a bat, they'll either be climbing or flying most of the time. They will move in these short, stuttering movements, and it'd be very hard to get the kind of data that you need from those kinds of movements. In a flying animal, the animals are moving at hundreds of times the speed that a rat would move. So they might be only in a firing field for less than a second. And then that's just, again, you're not going to collect enough data in order to be able to get that high resolution, to answer your high resolution question. I think there's a bigger question, though, here, right? And that's, aside from just the data and nitpicking whether or not these things work, which I think is an important thing that scientists do, if it did turn out that bats didn't have theta rhythmicity, how much can we actually apply that to our knowledge of the brain? Remember, the road that I've taken you down started with a guy who had epilepsy, a human being with epilepsy, and then we've gone into rodents, which are relatively closely related to us, and we've gone into bats, which are actually more closely related to whales than they are to humans. They've had a very different evolutionary pathway in terms of their navigation systems. Every single bat has evolved from a bat that was a primarily nocturnal bat that primarily navigated through discrete packets of information, echolocation. So I think that it's really hard to make the kinds of sweeping statements that people make in this field. So yeah, the answer to that is that it's complicated. But I think what it ultimately comes down to is that the scientists that are involved in this have too much invested in these individual theories, because we have now given the scientists so much financial incentive to have their theories proved correct, when what the goal of science is to try to prove theories incorrect. And so if your theory is proved incorrect, that would be the ruin of your lab, and that just leads to sloppy science. So this actually led to another project, and I'm going to talk about it very briefly because I think it's really interesting, and I have no idea what it means. So one of the really cool things about this alternate technique that I use for generating examining rhythmicity is that unlike the original technique, where we have to take all of our data and smush it together in order to get one number, I can conditionalize. I can say, based on how fast the animal is running, what does the rhythmicity look like? So this is what this looks like for this grid cell. So I've got on the y-axis the running speed, and again, this is the probability. The color here is the probability that another event happens within this window at about 100 milliseconds. And so again, we're seeing these waves. But because I've now exploded this over speed, we can see something really bizarre. These waves actually slope out as you go faster or slower, or get closer together when you move faster. And what this means is that the cell is actually oscillating faster with how fast the animal is running. Now, this is actually not that surprising. This is what happens with that global clock. The global clock runs faster when you're moving. And the thought is that because it's doing navigation, you have to do more navigational work when you're moving, so it's just running, the system's running hotter, it's running faster when you're moving. But that was not the only thing we found. We found some cells that did that, oscillated faster as the animal moves. We found some cells that seemed to not care about how fast the animal moves. And then we found these guys, which actually oscillate slower the faster the animal moves. No one's ever described anything that looks like that before. And I have no idea what it's for or what it's going to do. And I think it's a really interesting avenue for future research. So I was working on another project that has not yet come together, so I'm not going to talk about it right now. It's still actually being worked on in Boston. I have been very lucky to work with some very talented undergrads in my time. But after I finished that last project, there was a lot of chaos in my life. So I came out here in November. There was the Society for Neuroscience conference moves to different cities around the world or around the country, and I was in Chicago this year. I asked to meet with Dan Dombeck over at Northwestern, and I gave a job talk there because I was looking for a postdoctoral position, and I got a job offer. He gave me a job offer while I was out here. It was a really confirming thing, and it made me feel a lot better about my future, which was good because the next thing I found out was that we were pregnant when I got back from that trip. And then a month later, I found out that I was double pregnant. I defended my dissertation in March of that year. The girls were born in July, and then we moved here in August. So 2016 was a crazy year. And you might ask why I would try to cram all that in 1 year. Why was I so excited to get here and start doing some work in Dan Dombeck's lab? And the reason is that Dan Dombeck's lab is using some of the cutting-edge technology that we've developed as part of the Brain Initiative for looking at the brain. So, and I really like this story, actually, because it really points that to the idea that we have no idea where the best science is going to come from, the science that's going to make the biggest impact in the world. So here is a jellyfish. This is jellyfish, is studied by naturalists, by oceanographers. They get very little funding comparatively. This is a Victoria. This is a jellyfish that fluoresces green. It glows green under blue light. So some clever biologists figured out what protein that was, and it turns out that it's a relatively small protein, and a relatively small protein is really useful because we can put it wherever we want. We've put it in worms, we've put it in mice, we've put it in rats, we've put it in monkeys, we've put it in human cells. This has been one of the biggest revolutions in science because now we can just look at things in the brain and see where they are. We have now taken this protein and modified it. We can take this protein and stick it on other proteins and see where those proteins go. Or, in this case, this is Gcamp. We can break the protein in a specific way, bind it to other proteins, and make it so that it only goes green when it is in the presence of calcium. Now, calcium rushes into a neuron when the neuron becomes active. And this makes it so that we can collect some of the most incredible data I've ever seen. And I never get over seeing this data being collected in real time. So what I have here, this is from an animal that is awake, behaving. He's in a virtual reality world. So he's running on a treadmill, and he can see things moving past him. So we can actually get him to navigate through this virtual world. And I can look into his hippocampus and see the neurons in real time light up. So that is just blows my mind. So that's why I'm so excited to be here. So that's my talk on my science. And I'm happy to talk. I could talk to you literally all week about my science. So if you have any questions, please ask me. I do have my dissertation on here, so I could defend my dissertation if you want. But the other thing I really wanted to talk to you guys about was the state of science in our country and the world today, and why it matters so much that we take a stand for it. The March for Science, you were all hugely inspirational to me in terms of, because it has felt so long as a scientist that society has it against you, that society is working against the end goals of science. And the March for Science and seeing all of you get so excited about it really inspired me to think not about as if I'm being trapped in a collapsing science system, but more of these are the problems that science has, let's try to fix them. So this is the state of science today. Now I'm really going to focus on life science in United States because that's the field that I'm in, but other fields are also been drastically affected by similar phenomena. But life science is actually doing relatively well. So this is how much in current dollars, so this is just the dollars of the day, we have funded medical science. So Bill Clinton and the Congress in the 90s doubled the NIH's budget over a few years. So you see this increase in budget that happens until about 2004. And then we have not increased scientific funding at all. Against inflation, we've actually slipped. We are funding science less than we were 15 years ago. Now that's a problem. And the reason why that's a problem is that when you add money to science, it's like adding food to a culture. It's you're adding food to an ecosystem. We're going to make more scientists. We've trained lots and lots of scientists. The number of biology students has increased. The number of biology PhD students has increased. But the jobs aren't there. The money isn't there. If we look at how many principal investigators, this is the people who get the grants from the government that run their labs, if we look at how many there are, it flatlines when Congress doesn't increase funding. This also plays into the problem of the cost of higher education. So the cost of higher education really closely tracks cuts to funding for state institutions. And this has meant that all secondary or tertiary education educators have been feeling the squeeze. So a generation of scientists are right now being left behind. And I'm not saying this just because I'm a young scientist. Neuroscientists have it good. In the Brain Initiative, moved a bunch of money because there was no extra money for it. They moved a bunch of life science money into researching the brain. And the technologies and the subfield I'm in, I'm going to be okay. This is not about me. This is about science in general. This is the number of graduate medical students over the last since the 1970s. It's just increased. This is the number of people who get grants that are 35 and below. So you work really hard until you're 35 years old. You try to get funding from the government, and then you can't. So in 1982, 16% of the applicants were under the age of 35, or the recipients were under the age of 35. Now it's only 2% because you have to have had so much success already before you can get compete for this limited amount of funding. In the same breath, scientists aren't retiring anymore. Scientists are working they don't retire, they work until they physically can't anymore. And the result of this is that more and more grants are going to older applicants, and there's no new jobs for people moving up. This causes a network of pressures in science, in academia, and I'm going to talk to you about a couple of them. I'm going to come up with a more comprehensive. There's a more comprehensive list too, but I'm going to focus on the things that I think I can have an impact on, and I'm trying to have an impact on. So this is a great cartoon from this red pen, black pen. This is nature. This is one of the big. This is a cartoon of nature. This is a parody. And it was done after we saw there was a really high-profile study that was really followed by the news, where we created, we had a new way of making stem cells. We could make stem cells without destroying embryos very easily and cheaply in the lab. And it turns out that these results were completely falsified. The funding pressures on scientists mean that if you don't get published in these high-profile journals, then you will lose your job, you will lose your livelihood. A lot of these people have no skill sets outside of science, and the industry jobs aren't there. And so they haven't been given, if you have been told until you are 30 that you're gonna have a job, and then suddenly the ground falls out of below your feet, what happens, right? And this has happened in a lot of fields, but what's been happening is that this causes a pressure on scientists to get results that are extraordinary, so that they can publish in these high journals. So it's not hurting the scientists as much as it's hurting science. There's been another phenomenon that's been called the reproducibility crisis, and I think this is something that should be covered in the news more, but it's very difficult to talk about it because it makes it sound like science is wrong. This is actually a research article that's been cited over 1,000 times now, where they looked at the statistics that we run in our papers and the way we design our experiments. And we are disincentivized for publishing negative results. You shouldn't publish negative results. You'll never get into a journal like Nature if you are looking at results that don't show something new. And what this means is that there's a bias towards selecting for things that are statistically significant. And this means that we're wrong more often about that number, whether it's significant or not, than we think we are. And so most papers contain at least one thing that is probably false. Now that's very troubling as a scientist, because I trust science, and I trust scientists to tell me, or I trust other scientists to do experiments that are going to inform my work and inform the way our society works. There's also a big problem in education and what happens to people when they start teaching people. So this is Mary Votyo. I believe I'm pronouncing that right. She was a adjunct professor at Donetsk University. She made less than $3,000 a year when teaching a full course load. She did not have health care. She was laid off after she found out she had cancer and died outside of her apartment. This is actually a shockingly common story for adjunct professors. Adjunct professors are just completely abused. They've never been able to unionize, and there's been more and more pressure on universities to slash costs for education because they're getting less funding. It's often people have often talked about hiring celebrity professors or paying professors too much as a cause for the increase in cost of higher education. Professor pay has not gone up dramatically. Even building costs haven't gone up as dramatically as people have said. The real cause is the cut of state funding. And it's trickling down to people that, I know someone who was an adjunct professor, and he had a lot of trouble for a while. So these are just some of the problems that are facing science. There's a lot going on. And here are a lot more of other problems that I think are a big issue right now. But it's not all doom and gloom. I think we can do something about this. We can move forward and fix these problems if we accept that they're problems, bring them to society, and say we're going to work together to fix them. And so the two things that I'm really focused on are the reproducibility crisis and the loss of public trust. This is something I go at with my career and with the research I'm doing. You may have noticed that in the second talk I talked a lot about verifying the results of another paper. I think this is something that we really critically need to do these days, and we need to start funding that more. And so I do that in the course of my research. I think all of these things feed into this the loss of public trust. There's been an idea that scientists are part of some conspiracy. The science is a liberal conspiracy, is a thing that has been tossed around, that the academics aren't part of real America, this kind of thing. And this is very troubling because we want to have a constructive dialogue with the public. We don't want to be alienated and up in our ivory towers and not be part of the world, because we depend on the world for our funding. And so one of the things I think we can do about this, though, rather than try to tackle all these problems at once, is to try to get out in the world and try to reach out to people that don't normally talk to scientists or that don't normally hear what science is going on. So this is a brain fair that happened just a couple months ago. The Northwestern neuroscience graduate students organized this whole thing. I'm super impressed with them for doing it. We did something like this with the Boston Museum of Science when I was in Boston, but it was not nearly as big as this. They basically took over an entire high school, set up 25 odd stations, all with neuroscience science experiments. I had a blender that had some sheep's brains in it. There was a setup where they were recording from neurons in a cockroach's leg, which you can actually buy a kit for online. That's like only $20. I recommend everyone try to do it. But the other thing they did was they reversed it. They reversed the signals so that they could play the leg, so they could make the leg kick. And so they played some Rihanna, I believe, into it, and it started dancing. So it's just kind of really fun, exciting things where you can start to talk to people about things like the experiment where I was blending up brains. I was talking about what a brain is made of. It's made of mostly fat, because fat is how your brain insulates things. And so that's something most people just don't know. And so we were taking the fat and extracting it from the brain and showing people, look, this is all fat that came out of a brain. So these kind of things are really great, but I think they have a problem in that the kinds of people that go to these things, these kind of fairs that go to a science museum, they're already biased. They're already people that have interest in science. They're like a lot of the people in this room probably. And I think we really need to cross over that threshold. And I don't really know a great way to do it. But one of the ways I'm trying to is with this YouTube thing that I've built. So I actually started this the day before the March for Science. I got inspired. I was sitting in the car. I thought of an idea for a bioreactor I could build in my house. And then I just did it. I took a bunch of footage. I slapped it all together. It's not really well edited as I'd like. But, it's the first step, I think. And I really want to keep moving forward with it. But the main goal is to bring science experiments to people that they can do in their own homes, that they can physically relate to, and to bring scientists to people. So here is just a brief clip of this video where I'm just talking about the experimental design. So I had 3 chambers where I was trying to create gas using biology. I've got those collection spouts we made earlier. I'm going to carefully label. I've got 3 collection chambers here, the bottom of where I'm going to collect my gas, and I've been collecting gas for about a month now because these bioreactors can be slow. And now the fun part. I've got this guy. This is my control bottle. I'm going to take the lid off. I'm going to take one of those downspouts I made earlier, screw the lid on, and now, if this reaction makes any gas, it's going to go through the straws and out the end here. And now, if I gently place that end under, now this is the control bottle, if I gently place the end under the mouth of the bottle that I'm going to collect the gas in, the gas will bubble in there, and then it'll just be waiting for me when it's all done. All right, so that's enough.
And
of that.
finally, our termites. The termites.
So here's a bit of a preview of what I'm planning on doing next with this. And I'll talk about this a little on the next slide, but I need more help doing this. I've realized it's just making video is very difficult and very time consuming. But I fortunately have a lot of people that are interested in helping. So I'm thinking the next time around, I'm going to have more help. And if you're interested in helping, please let me know. I am looking for hands all the time. But what I'm going to do next is I'm going to take the gas that I've collected, and I'm going to put this balloon into a bottle. So the gas that I've collected will be on the outside of the balloon, and then the balloon will be just sitting snug and wrapped up in all that gas that I've generated. I can put a little warm water in this balloon, and then what I've got is I've got a little model Earth. It's like a little ocean in a bottle with a little atmosphere, and I'm going to stick that in some cold water, and I'm going to see how long it takes for the whole thing to cool off. And hopefully what we'll see is that the different gases cause the same amount of water, the same temperature water, to cool at different rates. That's what we're going to try to do next. So what have I learned through this whole process? So the first thing is that making this kind of stuff is really time consuming. I knew this. I actually did some film stuff when I was in high school, and my cousin is a director out in California. But I really, it just takes up a lot of time. And so I think you should always ask for help. And one of the things that I've been really surprised by was after I started doing a lot of this myself, was that so many people are willing to help. But you also have to realize that not everyone can commit all the time to it. So you have to be able to adjust and be flexible when you're starting this sort of thing. One of the really exciting things that came out of it, though, is that a lot of the people who did watch this were people that I know and trust as scientists and engineers. And I've started getting comments from people, and so I'm actually starting to get sort of a peer review process going on with my own videos. Now, admittedly, I handpick my reviewers, so that does make me pretty biased. But it's a great way to get another set of eyes on the experiment, and I'm actually really excited to sit down and talk to one of the people who's given me a lot of feedback when I go back to Boston. So I'm going back to Boston over the 4th of July week, and so I'm actually planning on doing the next round of this experiment when I'm in Boston. So I'm gonna since the gases are taking so long to gather, I'm gonna do some chemistry to generate the gases more quickly, and I'm going to shoot some follow-up experiments. My cousin from California is going to be there, so I'm going to get him to do all the videotaping and everything, so that'll be good. But another goal that I've had for a while, and I'm still trying to fight for it, is I want to put together a slideshow of photos of the Ethical Humanist Society and other pictures I saw from around the web of the March for Science. I think that it'd just be a nice thing to have on the channel. Another big thing I want to do more is more interviews with scientists. I think a lot of people don't know the scientist's voice. They don't hear people talking about different stages of their career, what being a scientist is like. So actually yesterday I sat down with a graduate student from Northwestern. He brews beer, which is actually really in line with my whole bioreactor thing. But he also does neuroscience fruit fly research. And so we had a really interesting discussion yesterday. I'm going to have to cut out like 99.9% of it, but I'm hoping to put together something that will show people more about what it's like to be a scientist and what we think about. So I've been doing this for a while, and I have a lot of people to thank about a lot of different things. This is the lab that I worked at in Boston, and really my PhD wouldn't have happened without all these people. This is, again, my PI, Michael Hasmo, who I'm extraordinarily grateful to. He has a reputation, well-deserved, as being the nicest guy in neuroscience. I also have been lucky enough to be supported by a lot of grants, including my own grant. And I also wanted to say this is dedicated to my grandfather, who passed away a few years ago with Alzheimer's disease, and was one of the inspiration for me to start studying the brain and also thinking about science in general. The animals. This is actually this is a tribute that in Germany to research animals. I think it's important to have respect for the animals that you work with, and because while I think the work that we're doing with them is necessary, I think it's important to consider that there is a moral cost to what we're doing. And finally, I'd really like to thank my brothers and Siobhan up in the balcony. Siobhan is an excellent teacher and writer, and I really couldn't have done any of this without her unending support. So thank you, Siobhan. And with that, I will take questions.
It's a marvelous talk. For people who are interested in learning more about the social challenges of science, the epistemology, why we have so many debates about it, where would you suggest that we learn more?
So this is something I think there's not as many resources out there as there should be, but there are organizations that are trying to do more to do that. So actually the March for Science has become a movement. It's become a social organization itself that is pushing towards this kind of thing, and it's still very much in its early days, and it's something that a lot of people are talking about. But there's also the American Association for the Advancement of Science. They've done a lot of really great work to try to bridge these things, but I think it really is a thing that we need to expand on as a society.
Jason over here. I have a question on the reproducibility crisis. I didn't quite follow why there is one. Are the peer reviewers just not doing it? Can they not do it? Or, I mean, to me it seems like that's the fundamental part. You see if you can repeat redo the experiment and see if you get the same results.
That is a big subject of debate, is why this seems to have become such a big problem recently. And basically everyone agrees that one of the biggest problems is that no one is trying to reproduce experiments. People are doing it in the course of your research. Like if I want to expand on someone else's experiment, then I will redo that experiment to make sure I'm doing it right. But there is a lot of disincentive to spend resources on that kind of research. The reproducibility crisis really flared up after a psychology study that was really well reported in public media. It turned out the scientist who was doing those experiments, when he was trying to reproduce his results later, found out that his results wouldn't hold up. And he came out and said, look, this just didn't hold up later. And this led scientists to say, well, hold the phone, we've built a whole field on this idea. So what are we going to do now? And there's been a movement, and so I briefly... So there's a reproducibility project, which their website is here. So this is a collection of scientists that are really interested in making sure that we do science in a reproducible way. And so one of the things that really contributes to the lack of reproducibility is how hyper-competitive science has become. Because now we're at a point where we don't want to share our data. If you share your data, that means that you are less likely to get as many publications as you can out of that data. And that means that you won't be able to get funding, because it's such a competitive thing that one misstep in your career will cause your whole career to collapse. And so they've been pushing for a movement among scientists to push for open science, so that when you publish a paper, you publish all of your code, all of your data, everything that anyone would need to see what you're doing. They're actually even pushing for things like you design your experiment and you publish the design of your experiment before you do the experiment, and then you publish the results. And so that way you don't get the same bias to saying just positive results get published. But there's been there hasn't been that much change at a whole system level. I think it's going to be slow change to actually react to this appropriately. And I think that there's a big problem in that as well, is that the way we disseminate science to scientists is through these large journals like Nature and Science and all of that. And this is behind a paywall. You have to usually get access through an academic university to get it. So there's a lot of money involved. And so a lot of they really shape a lot of the policy about how these things happen. And it's really, they are making changes. Nature has made a lot of changes to its submission process to try to make science more public and more open, but it's still part of the problem is that we just can't get the science out to people, and we can't get the data out to people in order to verify the results.
Yeah. Hi, Jason. Are the problems with science specific to the U.S., or is this a worldwide crisis?
So I think there are a lot of problems in the U.S., but this is a worldwide crisis. But the thing is, America is such a leader in science. All of the biggest universities in the world are in science. All the biggest research money is in the United States. The United States funds more research than any other country. And this means that when we have problems, it's a problem for the whole system because science is an international community. So this is I think it's an issue that spans globally right now.
Hi, could you give a definition for dementia and for Alzheimer's as being a subset of dementia, and what you see in scientific research for the future of those topics?
So dementia is usually a degenerative disease of the brain that causes profound problems in terms of being able to maintain a normal life. Alzheimer's specifically is caused by a buildup of a protein called, we believe it's caused by a buildup of a protein called tau or A beta. There's somewhat of a battle between those two proteins in the field. But it starts building up in the parts of the brain that I study. And this is why it affects memory, is that it starts building up in those areas, degrades those areas, but then it moves out. It moves out to the rest of your brain. And then, so it causes much more bigger problems than just memory problems. And so right now, there's been a ton of money poured into these fields because, but not, I don't think enough, actually, because Alzheimer's is going to be such a problem for our society. We are when the baby boomer generation really gets into the full force of having of Alzheimer's disease and other forms of dementia, we are just not going to have the resources to care for all these people. And research on trying to fix this part of the brain has been frustratingly slow. Part of it is that other diseases like ALS, we'd be able to track that down to like one mutation that causes that, but Alzheimer's seems to be more complicated. There seems to be a lot more going on. For a long time, we were chasing down a red herring. There was a hypothesis about losing a particular neurotransmitter involved in memory, and we got stuck on that for a while. And still, a lot of the drugs we used to treat came out of that research, Alzheimer's disease, as ameliorative drugs, but none of them actually stop the progression of the disease. There are some things in clinical trials now that actually attack the proteins now that we think are misfolded that are causing the problems in the brain. They're in clinical trials. There's a lot of hope for it, but they're also, so I'll say I'm cautiously optimistic. I have a lot of genetic risk in my family for Alzheimer's disease, so I try to look at Alzheimer's research always optimistically, because it can be really scary. I can't. My grandfather had Alzheimer's disease. His brother had Alzheimer's disease, and my grandfather he worked for Bell Labs. He was one of the people that invented the transition between having to call an operator to make an outgoing call and having phones and multiple phone lines in a single building with rotary dials and everything. So he was a brilliant engineer, and then he got Alzheimer's disease, and watching him slowly lose everything that he identified as himself, it was really heartbreaking. And it's such a painful disease, and it's so costly to work with. I think it's something that we really need to make a priority. But in the same breath, I don't know that we understand how memory works in general or the system works to really say that we're going to pin it down right away.
Thanks for coming out and giving the lecture for us. I am wondering, you made a comment that if a lab comes up with an incorrect result, they lose their funding. So I'm wondering where the funding comes from, and I understand that the pharmaceutical industry provides funding, at least for some research, and do you think there's any problem with pharmaceutical companies wanting to get certain results that might be playing into this problem?
So first of all, I want to say that it's not that people get incorrect results to lose their funding. It's when people get negative results. It's so if you're trying to see, you say, my hypothesis is that I'm going to see this when I do this experiment, and then that doesn't happen, well, then usually there are two explanations for that. One, you've done the experiment wrong, and two, that the thing that you're looking for isn't there. And more often than not, there's so much disincentive to actually say that this isn't here, or this is evidence to say that this isn't here, because it takes a lot of evidence to actually show that people usually move on to a different problem rather than go after it, because you just can't get money if you're not going to get positive results. So so most of the funding in my field comes from the NIH, the government. You'll apply for grants from the NIH. You back up your ability to do science with your ability to get papers published through the peer review process. So that's usually the biggest factor on whether or not you're going to get funded, is whether or not you have a list of papers saying, I am a successful scientist and I can do science, so you should give me more money to do more science. But other things play into it as well. So there's a big process that happens. So scientists are actually involved in the process of choosing which group gets funding. So the people will get a stack of applications. Professors will get asked to go to Washington, D.C., read all these applications, and then they discuss them and score them and figure out which ones that are get funded. But we've shown that it's become so competitive, and there's so many applications that after there's a certain quality bar that you have to reach before you might get funded, and then after that it's basically random chance. Which person gets your grant? Did they like your particular project over another project? And people are so highly qualified that you end up having to pass over excellent grant after excellent grant after excellent grant before in order to keep the system running. There are some private agencies. My lab now is funded by a lot of private grants that do provide some funding. That is like a no strings attached type of funding, so not the pharmaceutical industry type of funding. And there's also research that happens in the pharmaceutical industry and that is funded by the pharmaceutical industry. But I think it is a real concern. You have to, that the pharmaceutical industry is gunning for particular things. We've actually shown this. People have done meta-analysis of papers based on where they've got funding, and it dramatically affects what hypotheses you're going to say are true. The hypothesis they want you to test is usually, does my drug work? And so they're going to pay people until they can get that data. And so that's a real concern in the field right now. And that's one of the reasons why, as a society, we should band together and say we want to fund basic science research that has no goal of making a product that's going to make money. And so that's really where I think the role of money coming from the government should play.
I think we have time for one more question. We're running a little late, but we're going to do one more question and we'll finish up for the day.
Thank you, and we're delighted that people like you becoming members. You're changing our whole place and bringing life, so I thank you, and I thank your wife and those kids. It's great. As a science teacher of 20 years, I don't know how to answer this question. I don't know if there's an answer. When I was a kid, I think Descartes was still, no, when I was a kid, I could understand Scientific American. I could understand popular science. Young kids now, science is so incredibly complicated, and it's so scary. So what do we do? How do we encourage young kids who don't have IQs 193 to go into science?
That's an excellent question, because I know a lot of scientists, and I do science, and yes, science is really complicated, and if you want to come up with experiments, you do need to learn a lot. But the vast majority of my job is pushing buttons in a windowless room. It actually, if you want to get into science, anyone can do it. Anyone, because there's just a lot of physical work that needs to be done. Right now, the way we've structured the funding for science is that anyone who is smart wants to get into it. There's such a societal need for science. And if you have the skill set to do it, there's a lot of drive to want to do it and to get involved and do it. And so we're at a condition now where, because of the lack of professor positions, the positions that you really have to be the top level to get to, we have people who are working as technicians in laboratories that are primarily doing like ordering equipment, cleaning things, that kind of work, that they're getting research experience. And so to get that research experience, they are extraordinarily competitive. These are kids with bachelor's degrees that did undergraduate research for years, that are on published papers, and they're doing a job that isn't skilled labor. We have seen an increase in other positions that are not positions that are funded. And so I think that this is not just a problem in elementary education, this is happening in all levels of science, that it's just really competitive to get into it. And we need to make it so that people can do the physical work of science without being highly trained academic scientists. I think this is a big thing. So there's been a lot of talk about trying to make more career scientist positions, so make it so that the goal of being a scientist, this is the way we target the goals for science is like saying you want to become an engineer and you want to work at someplace great like Google. Well, most people will consider that like a really huge success to be an engineer at Google. But instead of that, what we're saying to scientists is, oh, well, you're not successful unless you can't get financial success at this unless you become a upper management level position at Google, right? And that's just not a sustainable model. But I think reproducing science is actually an opportunity to really bring more kids into it. I think we have a lot of science that isn't this kind of highly complex, cutting edge, like really trying to invent new technologies and that sort of thing, which takes a lot of interdisciplinary knowledge, which makes it really complicated. We've got a lot of experiments that should be done, that should be redone, that we don't currently fund to do, and I think that would be a great opportunity to get more people involved. And so I hope that society can move towards that, but that it will take lots of money and space and resources and goals to get to that. And so.